up, guys? It's Jacob here. If you're anything like me, then you are a little lost when it comes to the cell and how it replicates and how it makes proteins. I just tend to get really confused really easily. So I'm going to break this down into 4 sections and hopefully it'll make a little more sense. The cell goes through 2 different things with its genes, with its DNA. First of all, it wants to replicate. So the cell wants to make more DNA to be able to do replication and make a new cell. But also the cell wants to make proteins because proteins do everything in your body. They are the enzymes of your body. So the cells also want to make proteins. And so let's go ahead and go through those 2 topics. But we have a difference in eukaryotes and prokaryotes. So eukaryotes are like human cells. They are our cells. And prokaryotes are things like bacteria. All right. So for DNA replication, our cells are linear. They are linear DNA. And I'm going to draw a little DNA here. Here we have hydrogen bonds between T's and A's and G's and C's. G's and C's have 3 hydrogen bonds in those base pairs, and T's and A's have 2 hydrogen bonds. DNA replication is just making a new copy. We want to make a new copy of this DNA. So we want to make another one. And we make another one using lots of enzymes, one of which being helicase that will unzip it. Then we have polymerase 3 that will put on new base pairs. And our ending product is going to be, well, another DNA. That's what we want. So how is that different from bacteria DNA replication? Well, it is pretty similar. However, bacteria have circular DNA. And oftentimes it'll be double-stranded. So here's a double-stranded DNA. And I'm not going to draw all those out. But it also wants to replicate. The only problem is when it's done replicating, it's going to have a new circular DNA. It's actually going to be attached inside like this. It's going to be a ring, and it's going to be like this. So we'll have an enzyme come and break those. But either way, the whole idea here is that replication just produces a new DNA strand for that cell, and then the cell can break and make another cell. Protein synthesis. This is where we hear terms thrown out like mRNA, and go ahead and write mRNA, and tRNA, and rRNA. So what is protein synthesis and how do those mRNAs and RNAs come into play? Well, I'm going to go ahead and draw a chromosome here for a human cell. On this chromosome, we might have a little piece right here that codes for a gene. Maybe I want to make an enzyme, the enzyme helicase. Well, I need to make more of them. So this little piece right here is going to code for the gene. This is the gene that will code for helicase. So what happens is this DNA opens up and we only copy this little piece right here. And we copy it using mRNA, ribonucleic acid. So we use a ribose sugar. So that will produce a little strand of this mRNA, just that little gene. Now this little gene is going to get cut up and processed, so in the end it'll only, maybe we'll cut out some unimportant stuff like here to here, stuff we didn't use, and it'll end up just being this little piece of mRNA. Perfect. And then this little piece of mRNA leaves the nucleus and goes and finds a ribosome, a little ribosome like this. And this ribosome is made up of rRNA. And then we'll have a little tRNA, and this will come up and it's a RNA strand that's hooked with an amino acid. Okay, amino acids. And we're going to have a bunch of them come up on here, hook in there, and eventually we'll form this long chain of amino acids. And this long chain of amino acids is going to fold and form a protein. That's the whole point of this, is forming a protein. Okay, Now the main difference for bacteria cells is we will have a little piece here that we want, and this will be the gene, but this one little piece is going to be already ready to go. So this mRNA, when it comes out of here, it's going to come out and, maybe I'll draw a little arrow, come out and be ready to go. And this can code for multiple genes, or excuse me, multiple proteins. We might have a start and stop, start and stop. So we might have a bunch of proteins made out of this, a bunch of smaller proteins. But nonetheless, this goes and hooks onto a rRNA, a ribosome, and then tRNAs have amino acids hooked on, and they'll go and put themselves in that ribosome, and then they'll end up making a long chain, again, of amino acids that fold and make a protein. Alright, hopefully this video was helpful in just giving you a quick overview, and hopefully it makes a little more sense now. Anyways, good luck. If you have any questions, let me know, and I'll probably make some more videos going into a little bit more depth on each one of these. Thanks, and have a great rest of your day.